Today, we share the planet with roughly 1.3 million identified animal species. But if you step back 875 million years, the Earth was unrecognizable. There were no forests, no insects, and no predators, just barren rock crashing into a dark, lifeless ocean. For the single independent cells drifting in that vast water, survival was a strict numbers game. A solitary cell faces a hard physical limit on how much food it can catch, how fast it can move, and how well it can defend itself against environmental hazards. The evolutionary workaround to that isolation was cooperation. Single-celled organisms, known as choanoflagellates, began physically linking together into colonies. By sharing resources and operating as a single unit, they drastically increased their odds of survival. This diagram compares a free-floating choanoflagellate with an identical choanocyte cell inside a sponge. As the first true animals, sponges operate as cooperative water filtration machines. By beating their flagellum, choanocytes create a current to pull in and share food. While cellular teamwork solved the problem of isolation, it created a new one. A sponge is permanently anchored to the seafloor. To actively hunt, run, or explore, life required a completely new architectural blueprint. That new blueprint arrived through the development of true tissues and bodily symmetry. Early organisms like jellyfish, belonging to a group called cnidarians, evolved specialized nerve and muscle tissues, granting them the ability to actively contract, swim, and sense their immediate surroundings. The design was then upgraded with the mesoderm, a third biological tissue layer that allowed for the construction of complex internal organs. Crucially, animals began clustering their nervous tissue and sensory organs at one end of their bodies, a process called cephalization. Life had invented the head. This illustration captures the Cambrian explosion. With new complex organs decoyed, the ocean devolved into an intense arms race. Prey, like hallucigenia, grew defensive spines. In response, massive predators like the anomalocarat evolved grasping appendages engineered to crack those defenses. The combination of heads, forward-facing senses, and biological weapons turned the oceans into a highly lethal war zone. To survive, certain lineages were forced to look toward a totally empty but highly hostile frontier, the dry land. Escaping the water meant facing three physical traps. Without buoyancy, gravity crushed soft biological tissue. The atmosphere immediately evaporated water out of unprotected cells, and aquatic gills collapsed under their own weight in the open air, causing suffocation. The biological engineers that successfully bypassed all three of these traps belong to a superphylum known as the Ecdysoa, the arthropods. This fossil of an ancient trilobite displays their primary survival technology, the chitinous cuticle. This rigid, segmented exoskeleton provided the physical scaffolding required to stand up against gravity, while acting as a waterproof seal to lock cellular moisture inside the body. But wearing a rigid suit of armor introduced a fatal vulnerability. The exoskeleton cannot expand. To grow, the arthropod must periodically split its armor open, crawl out, and wait for a new, larger shell to harden, a highly dangerous process known as ecdysis or molting. To solve suffocation, terrestrial arthropods abandoned gills for the tracheal system. Air enters through abdominal pores called spiracles, traveling down branching tubes into deep tissues. By routing oxygen straight to the cells, arthropods generated the massive metabolic energy required to run and hunt. They eventually repurposed segments of their thorax to support thin, membranous wings, making them the very first life forms capable of powered flight. By engineering a waterproof skeleton and internal direct-to-cell lungs, arthropods conquered the surface. Because of these distinct anatomical technologies, they remain the most abundant and diverse group of animals on the planet today. Our own evolutionary lineage, the deuterostomes, faced the exact same physical bottlenecks when we eventually made our own push onto land. Ancient lobe-finned fish utilized thick, rudimentary bones inside their fins to drag their heavy bodies out of the shallows. Over millions of years, these structures adapted to bear weight, evolving into the limbs of early amphibians. The final tether to the ocean was cut by the evolution of the amniotes. Reptiles and early dinosaurs developed eggs enclosed in protective, moisture-retaining shells, allowing them to reproduce entirely inland without their embryos drying out. That reptilian dominance held firm until 65.5 million years ago. A massive extinction event at the end of the Mesozoic era wiped out the non-avian dinosaurs, clearing the top tier of the terrestrial food chain. This evolutionary timeline illustrates the global reset that followed. Notice the severe pruning at the 65.5 million year mark. With apex predators abruptly removed, small, historically marginalized mammals rapidly diversified, claiming newly emptied ecological niches and drastically increasing in physical size. Over the course of the Cenozoic era, that explosive mammalian expansion laid the biological groundwork for the emergence of primates and ultimately human ancestors. From the microscopic rhythmic pumping of a single choanocyte to the dense neural complexity of the human body, every modern animal is a walking, breathing museum of the radical biological engineering required to escape the ocean.